telomeres, telomerase, and a fountain of youth theory. Presentation by Cyrus Negra. What is it? Okay, so our body is made up of trillions and trillions of continuously dividing cells. When they divide, they make a copy of their DNA as well. This DNA is tightly wrapped into structures called chromosomes, of which humans have 23 pairs. The problem is, DNA replication isn't quite perfect, and the ends of our chromosomes are often skipped. This can result in the loss of crucial and important genetic information that our bodies cannot afford to lose. To prevent the loss of this important genetic material, our chromosomes have something called telomeres on the end. Essentially, telomeres are repeating sequences of DNA which our body can afford to lose. In humans, the nucleotide sequence is a repetition of GGG, TT, A, GGG. As we can see, with each round of cell division, the telomeres continuously get smaller and smaller and smaller and smaller until eventually cell division must come to a halt. How does it work? On a molecular level, this is the process by which telomeres allow our DNA to replicate. Ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only expand from a free 3' hydroxyl. Replication machinery builds the lagging strand in a backstitching mechanism. DNA can only be added going 3' to 5'. RNA primers provide hydroxyl groups on regular intervals on the lagging strand template, whereas the leading strand elongates continuously in a 5' to 3' direction until the end of the template. As primers are removed and replaced with DNA where a 3' end is available, a space is left on the 5' end of the lagging strand, as DNA can only be added to the 3' end through DNA polymerase. As this process continues through multiple rounds of replication, we see the strands of DNA getting shorter and shorter and shorter. This end replication problem is solved through the enzyme telomerase. Ends of chromosomes contain a G-rich series of repeats, which we know as telomeres. The enzyme telomerase is able to recognize the tip of a repeating sequence. Using its own RNA template, the enzyme telomerase is able to extend the parental strand in a 5' to 3' direction. The enzyme telomerase adds additional repeats as it moves down the parental strand. This allows the lagging strand to be completed through the DNA polymerase alpha, which has a RNA primer as one of its subunits. By this method, the original information at the end of the linear chromosomes is able to get copied to the new DNA, meaning no genetic material is lost and our body is able to preserve the possible needed genes. How was it discovered? Telomeres were first discovered in 1938 by German biochemist Hermann Muller while investigating chromosomal mutations in yeast organisms. But it wasn't until the research of doctors Elizabeth Blackburn and Joe Gall in 1970 where we discovered that in mammals and eukaryotes alike, telomeres were made up of an almost never-ending repeating DNA sequence, TTAAGG. The abundance of telomeres in a variety of species allowed Blackburn and Gall to infer the importance of telomeres, which has led to the excessive research even at the present day. The work of Hermann Muller, Elizabeth Blackburn, and Joe Gall were all extremely influential in understanding the fundamental concepts of how telomeres work in our body. But the question of how do telomeres expand to prevent degradation was a question that riddled many scientists during the 1980s. In the 1980s, scientists knew that DNA polymerase could not copy the ends of chromosomes for DNA replication. Yet chromosome ends were still largely maintained in an organism. Additionally, they observed that the ends of chromosomes consisted of highly repetitive sequences that we now know as telomeres. Elizabeth Blackburn and her colleagues hypothesized that the cell must have a mechanism to add telomeres to prevent chromosomal shortening during replication. But the question that riddled the entire scientific world at the time was, what was this substance? It wasn't until December of 1985 when this question was answered. Scientists out of UC Berkeley, Elizabeth Blackburn and Carol Greeter, published a revolutionary paper titled Identification of a Specific Telomere Terminal Transferase Activity in Tetrahymena Extracts, which expanded on what the key missing ingredient in telomeres was. In the paper, the scientists performed an experiment on a substance named tetrahymena, which also had telomeres, but of the sequence TTGGG, unlike that of humans, which was TTAAGGG. AGGG. Due to the rapidity and synchronicity of the replication of tetrahymena, it made it the perfect subject for this experiment. The experiment went as followed. The scientists isolated the nuclei of tetrahymena, which included solely the DNA and proteins. They dubbed this the tetrahymena extract. They then proceeded to label all T and G nucleotides with radioactivity. Nucleotides were incorporated into the new telomeres and radioactively labeled as a new sequence. Scientists then proceeded to mix the tetrahymena extract with the radioactive T and G nucleotides along with a single-stranded DNA template that was made to mimic an already existent telomere. The results of the experiment proved that the tetrahymena extract mixed with the radioactively labeled T and G nucleotides was sufficient enough to create a telomere. If either of the T or G nucleotides were missing, a telomere could not be produced. A newly formed radioactively labeled telomere was being added to the single-stranded template. The same conserved sequence of TT, GGGG over and over again. After multiple rounds of biochemical purification, scientists Elizabeth Blackburn and Karen Greider were able to pinpoint what the key substance responsible for expanding linear chromosomes was. Telomerase. What is the scientific importance? Further research done by scientists on telomeres and the enzyme telomerase has allowed them to expand and see the relation between telomeres and the aging process of the human body, which has been very beneficial in scientists understanding the fountain of youth theory and how we could possibly live forever. We need to learn more about telomerase and telomeres. Its importance in our genetic makeup and our aging ability is without question. As recent studies and research have also found that telomerase and telomeres are influential in diseases such as cancer, melanoma, and many other life-threatening diseases, and how the implication of possibly more telomerase in the form of anti-aging pills could prevent that and could also help people keep their youthful look and possibly live forever. What is the societal importance? Overall, telomeres and telomerase are two extremely necessary functioning components of our human body. As research continues on both of these subjects and molecular genetics as a whole, scientists and researchers aim to hopefully find answers to questions like why we age, how we contract disease, and if there is a prevention for all these things. The societal impacts of these findings would be extremely immense, as it would be in everyone's best benefit to find out how we age, how we can prevent disease, and how we could extend our lives a little longer. This would improve innovation in our societies, it would improve overall quality of life, and it would just help us learn not only more about ourselves, but our world as a whole, and how we interact as living things with each other.